Okay, we're gonna go and get some ice. To put my samples to start preparing, putting in the primers and the buffers. Getting ready to prepare my samples so I can put the DNA of the red algae. We have all these Hallemania species and we want to know which ones are different. So we're trying to see which ones are different. So we just added up until 82.
Yeah, big dye, buffer, and water.
Okay.
You will add the primers and the DNA.
Okay.
Okay. To each tube.
Okay.
Yeah. What's next?
Sequence reactions and putting in 6 microliters.
Oh, 54?
54 is the next
Continue with 71.
Okay. And I'm going to continue on
71, 82.
So sequence reactions of RBCl. So these are sequence reactions of chloroplast encoded genes. And now I'm inputting the buffer into each one of them. Just continuing to add the buffer into each one of them, 2.5 microliters. This is all part of the Red Alga Tree of Life grant, and I'm also a REU student.
So you're doing a lot today?
Yes.
All the Hallemanias?
All the Hallemanias.
Very good. And then another day you're doing the other vaccine.
The other thing.
Very good. So you already looked and took pictures of them too, as I know. The database and everything?
Of all the red algae.
Very good.
Put in the DNA and the primers, and now we're about to put into the sequence machine. I'm gonna go and put it in over here. I'm taking it out of the tray. Okay. Close it. Have the user's name. Then we're gonna go down and we're gonna cycle sequence. Right here. And start.
Okay. Ready?
And now it's ready.
And then what you're going to try? What is this?
You're going to wait 2.5 hours to cycle sequence. I just sequenced the RB cell, which is a chloroplast encoded gene of the Hallemania. So in the meantime, we're going to prepare the Cephedex columns to clean the reactions. The next step is to clean the sequence reactions with the Cephedex columns. And this is the Cephedex right here that's already made. This is the column that we need to prepare. So I'm going to make 82 of these.
Okay.
I'm adding the Cephedex here, 750 microliters into each one of these columns. Once we add in the Cephedex, we gently take off the bottom of it. And we take out any bubbles. You can see the bubbles forming at the bottom. We take those out. You take out the bubble. Okay. And you gently take off the bottom. And then you place it into the collection tube. And then afterwards, we put it in here. And put it into the centrifuge. We put it in for 4 minutes at 4,000 RPM. We just continue and repeat the process for the rest of them. There's 82 altogether. We just finished with the rest. Okay, we will take from out of the centrifuge. We'll discard the collection tube, and we'll place the column into the micro centrifuge tubes right here. And we'll just repeat the process for the rest of them.
Perfect.
Okay, I'm about to take the products out of the thermal cyclone machine. And once I take them out, we're going to go ahead and clean them. The DNA, taking it out.
No.
Okay, I'm taking out of the cycle sequence products. And I'm going to place them into here, but make sure that I do not touch the column. Put it directly in the center, make sure it's not touching the pellet. And I repeat the process for the rest of my products. And this is cleaning the samples. We're going to take it, put it in the centrifuge, close it at 4,000 RPM at 4 minutes. We'll take this out of the centrifuge. We'll trash the Cefidex, place this in here. The clean cycle sequence product is in here. We'll take it to the vacuum machine and we'll place it in there for 20 minutes until all the liquid is gone. We're placing the clean cycle sequencing products into the DNA speed vacuum machine. We're going to open them up so they can dry until all the liquid is gone, and it's going to run in here for about 20 minutes. It's been 20 minutes since we took them out of the speed vacuum. We're now adding high deformamide to resuspend the DNA. Okay, we waited 10 minutes, we vortex, and now we're adding the DNA that we just resuspended into the 96 well tray. Okay, I've just finished loading them into the 96 well tray. I'm going to place this on top and cover them. And then I'm going to bring them to the thermal cycler. I will place them in the thermal cycler machine, close it, and it'll denature for 2 minutes at 96 degrees. And I'll wait for 2 minutes. I'm getting ice right now to keep the samples nice and cold. We're taking them out now, and now we're going to bring them to the sequencer machine. We're in the sequencer room right now, and I'm checking for bubbles at the bottom of each sample because it might interfere with the sequencer reading it. Okay, I'm simply placing them into the tray, and then I will cover them with the cover. Snap it on. I'm at the sequencer, and now I'm checking to make sure there are no bubbles in the line. And there are no bubbles, so now I can load the sample, the tray into the sequencer. Okay, open it. I just simply place it in there. That's it. Okay, now I'm inputting information into the computer so that the sequencer can run, such as the polymer type and the capillary length. We have 80 samples. It takes 2.5 hours to run 16 at a time, so it'll take a total of 10 hours to run them all. Okay, now we're getting ready to open up the sequences, and we're going to edit it in the sequencer. I'm going to go ahead and delete the first part. This is the part that you can't see very clearly, so delete it, and then we'll go forth and start editing. Okay, I'm now editing the end. I'm going to go ahead and delete the last 20. I'm working on the RedToL. I'm focusing on the Halominaceae part. The blades and whatnot is the main part that I'm focusing on. Natalia has already did a lot of data, and I'm just kind of going ahead and finishing up the rest of the data.